colleague Bob Pelcovits from Brown University to give us a talk about the self-assembly of FD viruses. Bob.
Thanks, Mark. So yeah, thank you to the organizers for this invitation and for setting up this very pleasant and hopefully fruitful workshop. So I'm going to be talking to you today about the self-assembly of FD viruses. This is work done in collaboration with the group at Brandeis, which has this MERSEC grant for the past 5 years. And so, the theory I'll describe was done in collaboration with my graduate student, Hao Tu, and with Bob Meyer's graduate student, Nadir Kaplan, and Bob as well. And the experiments, which I think are the real stars of the show here, were all done in Zvonimir Dogic's group. And these are the people in his group who were involved with these FD experiments. Yeah, and Zvonimir will be here for the conference at the end of June to talk about the even more exciting or equally exciting work he's been doing on active matter. So FD viruses, just in a nutshell, what's special about these and why are they a great system for studying liquid crystal type physics? So here's a picture of an FD virus under an electron microscope. The thing to note is the length, which is, that's my pointer, nearly a micron long, so that you can really, image individual viruses. Relatively narrow, so that kind of puts it in this nice, Onsager regime. These things are fairly rigid, so it depends on the particular mutation you're looking at, but the persistence length, can be anywhere from 3 times the length of the molecule to, let's say, 10 times. As I mentioned, they're large enough that you can visualize individual viruses while at the same time looking at the aggregate behavior. These are also chiral objects, and you can actually control the chiral interactions, the macroscopic chirality, via temperature. But at the same time, you can genetically mutate them and actually create FD viruses which have opposite chirality. And then you can mix the left-handed and right-handed together and form achiral mixtures. If you look at these FD viruses in an aqueous solution, salted so that you get rid of the electrostatic repulsion, you'll find that they exhibit isotropic, holosteric, and smectic phases. And this has been studied, quite extensively by the people at Brandeis, probably 15, 20 years ago. Now, the excitement of the stuff that I'm going to show you, this particular self-assembly, has to do of seeing two-dimensional structures, which can be formed when you, in addition to the FD in the solution, you add a non-absorbing polymer depletant, such as dextran. And then via this depletion interaction, you form an effective, attractive interaction between the FD viruses, in addition to their hardcore repulsion. And in particular, if you pay close attention to monodispersity, and there are experimental tricks for doing that. If you make very monodispersed solutions of FD, you can form some really incredible two-dimensional structures. And so let me show you some of these structures just to motivate the theory that I'm then going to explore. And so I'll show you a few pictures now, and there will be more coming later on. They just every day I would show up at Brandeis, there would be a new observation of yet another structure. So the simplest one is just a flat circular disk. So I will show you a cartoon picture in a second of what's inferred about the arrangement of the FD viruses. But in short, what you're looking at is a top view of this disk. Has about 1 million FD viruses in it, about 10 microns across. It's flat, and the viruses, at least in the black portion in the middle, are all pointing out of the slide towards you. And then what this white rim is indicating is that there's actually tilting near the edge. But then there's more. I mean, if it were just the flat circular disks, that would be fun. But there's even more going on here. What they find, and I'll tell you, some of the conditions for producing this in a second, is that these things can actually spontaneously form twisted ribbons. And just to give you a sense of the scale here, the width of each of these ribbons is about 2 microns across. And then you notice there can be all sorts of junction points, and then these things can even self-braid. And then, as I'll show you a little later on, there are ways to manipulate these things using optical tweezers, which allow you to transition, from a disc to a ribbon. They can also use tweezers to, take two ends of a ribbon and join them together into a ring. There are all sorts of ways you can manipulate these things. And as I mentioned, before, there's even more, that I will show you. This is not the whole zoo of structures that they've seen. So, let me kind of summarize just in a simple, theorist-type way what they infer about what's going on in these flat circular membranes in terms of the arrangement of the FD viruses. So through a combination of 2D and 3D microscopy as well as electron microscopy, what they infer, is what I mentioned to you earlier, that in the center of these disks, the rods are well aligned, and then as you get near the edge, they are twisting over. And then when they look at this electron microscopy, they can see that we're not talking about a sharp edge here, but rather a rounded over edge. And in fact, really at the edge here, one has pneumatic order. Now, what is chirality? I mentioned these things are chiral, and I mentioned that you can have both left-handed and right-handed chirality. These in wild type, and then there's mutation Y21M. So, not surprisingly, what the chirality will do is tell you how, the sense of the twist near the edge. But what's also interesting is, originally when the experiments were done and we looked at this, we thought that, chirality was clearly the key issue. And that, if you had this twist near the edge, well, it was just because as you got near the edge, the molecules could adopt the twist that chirality wanted to encourage. In the interior, because they're trying to form this layer, the chirality was then being expelled. And this is back to the de Gennes analogy with superconductors. But interestingly, even achiral, when they make achiral disks, they have similar shapes. So we've begun to think that the shape at the edge, while having something to do with chirality, and certainly in terms of left versus right twist, chirality definitely plays a role. But if you look at an achiral one, you will find it rounded over as well. The difference will be in an achiral disk, it could be equally left-handed or right-handed. So now what's going on with the ribbon? Again, here's kind of a theorist cartoon just based on the results of the optical microscopy. So again, as you're seeing in the interior of the ribbon, any kind of twisting is expelled. And so you have these rods well aligned, but then as you go near the edges, things begin to twist over. But then, the structure as a whole has adopted this twisted ribbon shape. Now, let me show you a little bit more data, and then I can begin to turn to theory. So I mentioned earlier that the strength of the chirality can be controlled by varying temperature. And so that's what this data over here shows. These are actually experiments done in bulk samples. They're not actually measuring things in these 2D structures. But the assumption is that this is the chirality that one could then carry over to an analysis of the 2D structures. So as one goes up in temperature, the wave vector of the chiral twist, in other words, the inverse of the pitch, is decreasing, and hence the chirality is going down. Now, when they, so what are the conditions for forming flat membranes, disks versus ribbons? So, that can, that's seen depending on temperature, i.e. chirality, and the strength of the attractive interaction. So the concentration of this polymer depletant is a measure of how strong the attractive interaction is. And, what you see is that in this corner of the diagram, either low chirality or weak attractive interaction, ribbons will form, and, membranes will form up here. And I should point out that, actually you can't even see it in this picture, but for truly achiral systems, membranes will not form at all. Yeah, I think it's not, you can't see it in this picture here. So now let me give you a sense in just some simple way before going into any complicated theory as to, what the physical idea is in assessing membranes versus ribbons. So off in the right corner here, I've just copied that phase diagram you saw a second ago just for reference. And basically the idea is what you're playing off really is this chirality versus the edge length per unit area of the structure. And so if you're looking either at low chirality or high dextran concentration, you're going to form a flat membrane, a disk. And the reason being that, in this case, what you've done with the disk versus the ribbon is, you've packed a lot of these molecules in the center aligned with each other. You've frustrated the chirality, so that'll be fine if your chirality is low. At the same time, what you've accomplished by having the membrane versus the ribbon is a shorter edge length per unit area, which would then be favorable if you're talking about a high dextran concentration. And then, if you just invert these things, larger chirality or low dextran, then you can argue that a ribbon will form. But we also think that an important aspect of this ribbon formation, not only this play-off between chirality and edge energy, also has to do with the negative Gaussian curvature of this structure, as I'll tell you a little more when we get to the theory. So at this point, let me turn to the theory. So like I say, there'll be more experimental stuff coming, but this is a good point to introduce the theoretical approach we've taken. I should mention, so I'm going to be describing this phenomenological theory. There's also been simulation work that Mike Hagan at Brandeis has done and that I'm now getting involved with. But in this talk, I'm just going to focus on the work we've done within this phenomenological free energy. So basically, what we do here in large part is just assemble, well-known bits of liquid crystal and membrane physics using the Helfrich energy to describe these two-dimensional structures and the de Gennes Landau type free energy to describe the director orientation of the rods. And then in terms of the edge energy, everything I'm going to show you is based on a very simple model of just what's the edge length of the structure and then multiply that by some phenomenological coupling and call that the edge energy. But I should point out that Nadir Kaplan and Bob Meyer have done a more realistic model of the edge where they really consider the rounding over and the anisotropy and the surface tension. And so, they've taken a step beyond this very simple model. So now, just in terms of what goes in to each of these bits, like I say, the edge energy is very easy. The Helfrich energy is, the usual one. The only thing I just want to point out is this extra term, this higher order term, which ordinarily you wouldn't write in the Helfrich energy. And the reason, for adding that is kind of anticipating a result that I'm going to show you, is that when we look at this membrane ribbon phase diagram that I showed you experimentally and that we now try to give some theoretical sense to, we find that in order to have a reasonable description of the ribbon, reasonable in the sense of agreeing with experiment, this K bar, this Gaussian curvature constant in the middle term of the Helfrich energy, that has to be chosen to be positive, unlike lipid bilayers where it's typically negative. But a ribbon has negative Gaussian curvature, so this middle term in the Helfrich energy, will be negative in the ribbon state, and therefore, we need a higher order term just to stabilize the energy. Then, the de Gennes energy, we're just looking at it in the simple case of a single elastic constant. So this is just, Frank de Gennes, I guess more technically, the Frank free energy in these red terms in the one elastic constant approximation with chirality. That's all that's there. And then, to incorporate the effect of the, given the depletion interaction that these rods want to align with each other, we just add a phenomenological tilting energy with some constant C and just saying that, this favors parallel alignment of the rods with the normal to the surface. Okay, so in effect, this is really it. This is, what we're putting into our model. And, the rest of what will come, will be, some details having to do with curved surfaces. And so, Professor Ouyang has written a lot of nice papers that, help with this. So it's just a matter of setting up a local coordinate system on your surface with a layer normal. And here's the director making this tilt angle theta with the layer normal. And then one can write all the ingredients of the Frank energy as well as what's needed in the Helfrich energy in terms of all the differential geometry quantities. And those in turn all come from specifying a surface. So with a position vector for your surface specified, you can compute all of these quantities. The reason I've highlighted these two terms here in the Frank energy is because in the work we've done, we have taken a step beyond what's typically done, where the tilt angle, at least in previous work I'm aware of, is usually taken to be a constant. And as I've shown you from the experiments, given the tilting at the edge, it's critical that we allow for theta to have spatial variation. So these things go one small step beyond what had previously been in the literature. And then you take all of those ingredients and you just go back to the previous slide where I said the free energy was Helfrich plus Frank, tilt energy and edge energy, and just insert all of that, and that's the mess that comes out. But that's in principle it's a simple idea. It can get a little ugly with the details, but the basic physics, like I say, I think is pretty straightforward. So now, let me describe our approach that really kind of covers, everything that I think I will describe to you in the remainder of the talk. So, what we've done is the following. Within the context of this model, just that top line, we've then, turned to experiments and said, okay, what kind of structures are they seeing? So, for a given geometric structure, as I indicated on the previous slide, all of these quantities associated with the surface, the Christoffel symbols, the second fundamental form, all of that can be computed. And then what we do is we say, okay, given a particular structure, let's minimize the above free energy to determine the director field, but to also, in addition, figure out what would be the optimal geometric parameters of a given surface. And then what we can do, in effect really is two things. We could either, stop right there in the sense of saying, well, given some particular geometry, given this energy, what are the particular parameters of the surface we'd expect to see? Or, what we can do is carry this out, let's say, for a second geometric structure. For example, compare a membrane to a ribbon, and then ask, what are the relative energetics of those structures? So that, those are the that's the flavor of what I'm going to be showing you in the remainder of the talk. And let me just mention, since you'll see, various numbers and figures come up, that, what we've done generally is to use just dimensionless units where we're measuring lengths and units of the twist penetration depth, which experimentally is about 0.5 micron. So it's just back in that flat disk I showed you. It's basically just the size, the width of that angular white region, and energy in units of K, which is in real units like 100 kT. Okay, so now let me just give you a quick laundry list of the kinds of problems we've studied, and then I will go through some of the details. So, we began, Bob and I, just looking, not even, Bob doesn't like tensors, so he wasn't going to do differential geometry. So we began, just looking at the flat disks. And even within flat disks, you can ask, energetically, would you rather form a very large single disk, or would you rather, break up into smaller disks? And so, we've studied that, and the relevant result I'll show you shortly in the context of looking at the membrane ribbon phase diagram. But that's the kind of, those are the kinds of questions we addressed. Then the disk ribbon phase diagram, the four of us explored, and I'll show you the main results of that in a second. And then my student Howe and I looked at a number of other structures. So one thing I haven't shown you yet, the experiments, are these so-called rippled disks, which are basically precursors for forming twisted ribbons when you increase the chirality. So I'll show you the relevant experiment shortly and the theory we've done. Then another way to form ribbons is actually to grab them with grab disks with optical tweezers and stretch. I'll show you a very neat movie on that. And so again, we've done some theory along with that. and then time permitting, I will show you yet another structure that is formed when disks of opposite chirality merge together. And these particular structures have been named crenellated disks. That's Bob Meyer's name. We had been calling them cookie disks, and Paul Chaikin didn't like cookies and encouraged us to find a better name. So Bob, who's very much into Roman-type technology things now, came up with crenellated. So, this is just a kind of, like I say, a laundry list of what we've done, and let me, over the remainder of the time, give you some sense of the details. So, turning first to this membrane ribbon phase diagram. So, this is the result of doing the following. Let me, first explain without, there's no need to show a lot of technical detail, but at least explain, where did this come from. So, what we've done is just the program I mentioned earlier. We said we have, assume this free energy. Within that free energy, we can insert, any assumed structure we want in principle. And then we can, if we look, for example, at a ribbon, we can compare its energy with that of a disk. So that's basically what we've done here. And, these semi-infinite membranes just mean very large flat disks. And basically, the work that I haven't shown you details on, stuff I did initially with Bob Meyer, is actually to show that even if a disk is not truly semi-infinite, it could be, maybe even, 10, 20 microns in diameter. If you look at the behavior of the director as it twists coming in from the edge, it's a reasonable approximation just to use a semi-infinite geometry. So, we've tended to refer to these as semi-infinite membranes, but they really are, round, large, flat disks.
What is the dash line there?
Oh, yeah, I'm going to, sorry, actually what I'm doing, which maybe is not, I'm actually, coming to show you this. I'm giving a long prelude, yeah. So, let me now explain, what you're seeing. So basically what we did was, into our energy, we put in a ribbon, we put in a disk, and then, just for technical reasons in terms of ease of carrying out the calculation, what we did was just to look at varying the edge energy and the Gaussian curvature modulus. In other words, we did not vary chirality. That just would have been too time consuming. Even though, given the experimental stuff I showed you, you might argue, why didn't we explore chirality? That was just, it just would have taken too long. So now, here's what you're seeing. What you're seeing is this phase diagram, which at large edge energy, you see, disk formation. And let's see, I don't think I included this here. Right, if I won't bother going back to it. But earlier I showed you the experimental phase diagram, which, remember, again, at high dextran concentration, disks or membranes was the preferred structure. So, we see, not surprisingly, something along those lines. There's nothing, terribly profound there. But then, more importantly, what we find is, in agreement with experiment, a first-order transition between these large flat membranes and twisted ribbons. I'll come to the dashed line in a second. For now, just ignore the dashed line. I mean, I could really, could blank it out. And you'll note there are two slightly different solid lines here, one gray, one black. The only distinction between those was whether we used the Helfrich energy I showed you earlier, or if we replaced Gaussian curvature by pneumatic saddle splay energy. And you see there's very little difference between the two, which is not a big shock if you think that about the fact that the twist penetration depth is a half a micron. So in other words, the difference between saddle splay and the ordinary Gaussian curvature energy is only going to be in that very narrow region. So not surprisingly, when you get to the phase diagram, there's no big difference. Now, in terms of this dashed line, what that is there just to indicate that, so the dashed line is the result of the calculation that Bob and I did, and that calculation was to ask, when would you want to go from very large, or in other words, semi-infinite flat membranes to finite sized, still flat, circular disks. And that would occur down at this dashed line. That was the result of our calculation. So, the point of showing you the dashed line here is just to indicate the sense in which, this theory hangs together and agrees with experiment, again, at least qualitatively. That is, that if you're reducing this dextran concentration, if you're coming down in edge energy, you will have a first order transition. The theory says you'll have a first order transition, so does the experiment, from a large flat membrane to a twisted ribbon, and that will preempt what theoretically you could work out, as Bob and I did, to say that the semi-infinite membrane would become a collection of finite-sized disks.
As a practical matter, above the dotted line, your solution is the radius or size is infinite, and below it, it's finite, and 1 over that size goes continuously to 0.
Well, it's a continuous transition, correct. Yes, that's right. Yeah. Okay. Now, here's the so one aspect of the theory in terms of agreeing with experiment is that there's an additional prediction here which we think is important, and that comes back to something I alluded to earlier in terms of adding the extra term to the Helfrich energy. So I showed you earlier pictures of ribbons, and so it's straightforward for them to measure the ratio of the ribbon pitch to width. It's approximately 5. And what we find is that, again, within the context of our theory, it's critical that this k-bar, this Gaussian curvature modulus, be positive and approximately 0.15 over here in order to get a pitch to width ratio of that order. So, I mentioned that in lipid bilayers, the k-bar would typically be negative. If we put in a negative Gaussian curvature modulus, we end up with ribbon pitch to width ratios, probably 10 times larger than this. And so, the result here is that, we need this positive k-bar. Because the ribbon has negative Gaussian curvature, we have to add this stabilizing term to the Helfrich energy. But more importantly, this kind of stimulates, I think, thought, which we haven't yet fully carried out, as to why these kinds of systems should be different, let's say, than lipid bilayers, and why the Gaussian curvature modulus should be positive. But it's within the context of our model, a pretty clear prediction. So that I think is good news for the theory in terms of giving at least qualitative agreement with experiment. What's less and one other thing to mention that I said earlier is experimentally they do not see ribbons in achiral mixtures. The theory agrees with that. In other words, and which is somewhat non-trivial because remember we've put in this positive k-bar, we're encouraging the formation of a ribbon, a structure with negative Gaussian curvature. And so, while chirality will help that, it's not clear that chirality is absolutely necessary. But in fact, it is. If we turn off chirality in our model, this phase diagram, the twisted ribbons just disappear. But then, the one discouraging thing, probably having to do with our very simple edge energy, is if we then say, okay, here's a prediction for this phase boundary, and so how does this number, here it is in our reduced units, here it is in real units of kBt per micron, how does that compare to what's actually measured experimentally? It's actually, off by an order of magnitude. So that's, the one discouraging thing of the theory, and, may just have to do with the fact that we've used this very simple edge energy model.
How do they measure that number experimentally?
So that one they measure. So that was back in the. So you're asking how do they relate it? Sorry. Yeah, they're looking at the fluctuations, right, and measuring that, right, and then yeah. Okay, so now let me show you some additional experimental stuff that motivates more of the theory we've done, and so here is okay. So this is a very cool movie, where all they're doing is varying temperature. And so what's done, I'll play it maybe one more time, is they've started at high temperature, and remember high temperature means you've in effect switched off the chirality. Then they're quenching it, this is like I think 60 degrees C down to 10 degrees C, and so they quench it to low temperature, that's where you saw the ribbons form, but then if they reheat it, it's back to what you see here. So just maybe to show you.
The size of the depletants change or do not change with T?
Oh, over that, that's a good question. Yeah, there may be. I don't know how big a change. That's a good question.
They're not using the cleats that Arjun uses, that really do change size.
Yeah, so this again, I think is probably still dextran and not PEG. But yeah, that's a good question. I'm not positive about that. So let me just maybe show you this just one more time now that I've said the words. So they're quenching it. You form the ribbons, and then you can reverse it. And one thing I should show you again before moving on to the theory is, let me stop it kind of like right even here, or even back a little bit. As the thing you want to notice is that as these ribbons are beginning to form, we've kind of entered a state where the disk has this rippled structure. And so it's that idea of rippling that I want to go to next within the context of our theory. So, these are then referred to as rippled disks. So it's this structure. Here is a cartoon view based on a particular model I'll describe in a second. But here is the structure that is at least reminiscent of what I showed you, a moment ago. Now, one thing you did not see in the experimental movie, and in fact they couldn't, even if they changed to an edge-on view, it would be beyond experimental resolution, is that in the model I'm going to show you, which was actually suggested by Bob Meyer, is the disk is not only rippling in the plane, but also out of the plane. And the reason for doing that, why would you even think of doing that, is, again, because of negative Gaussian curvature. So, these pictures here, the top view and the side view, come from this mathematical model where, where we've got a radial coordinate at the edge which ripples. Little a is just the number of ripples, so here a is 5. And then, in addition, here is the rippling in the height, and the exponential is just saying that, experimentally, even if these height ripples are there and can't be seen experimentally, they're certainly, it's pretty clear they are tailing off, they're decaying as you go into the interior of the disk. So, that is just kind of the model that Bob suggested, and there's going to be, I've highlighted these parameters AZ and AR because those are going to be the geometric parameters that we'll try to, extract from our theory by minimizing the energy. But again, to motivate, why would you think of this kind of structure and rippling out of the plane in addition to in the plane? And that can be seen by plotting the Gaussian curvature of this structure. And what you find is that at these points, which remember, these ripples are where the ribbons eventually grew, those are points where the Gaussian curvature is negative and relatively large. So the idea is, those could be the seed points for forming the twisted ribbons. So, then the idea is to proceed, as I've indicated earlier, to say, here's a structure Bob made up. Let's put it into our free energy. Let's minimize the free energy and see what it tells us about those geometric parameters. We also get the director field out. These are kind of lousy. We don't have great graphics on this, so maybe I won't even show you more. Let me jump to the actual shape parameters, where there's really, I think, some good physical content. So what you're seeing here are these shape parameters: the height of the ripples out of the plane, the size of the radial ripples, and this is as a function of edge energy, which remember is just a simple way of saying dextran concentration, or strength of the attractive interaction. And so, as you go down in gamma, you are decreasing the strength of the attractive interaction, or the dextran concentration. And then for reference, so these gamma values are all in our dimensionless units. For reference, the dashed line, this value is where it wasn't a dashed line in my previous, it was those solid black and gray lines in the previous theory phase diagram I showed you. So it's where the theory predicts you would go from disks at high gamma to ribbons at lower gamma. That's the dashed line. And so the main thing to focus on here is that in the z direction, what the model is predicting is that there's essentially no change in the this an initial change from 0 to something non-zero. I mean, that's critical, that you have some negative Gaussian curvature, but then, beyond that, it kind of plateaus. And to make sense of that number 0.3, remember this is in units of the twist penetration depth, which is a half a micron. So this is 0.3 of a half a micron. It is too small for them to measure. And so that, if experimentally they say, well, the disks appear flat, it's actually consistent with this result. But more interestingly is the growth of the radial protrusions, which has a fairly dramatic rise as you go from the region where you have, where the theory predicts flat disks to where you have twisted ribbons. So, we view this as at least some, qualitative indication that this model is in agreement with the experiment, saying that as, in this case, we're not driving things with chirality again, given, that just technically is harder for us to do. We're driving things with the edge energy. But the idea is to drive the system towards the ribbon part of the phase diagram. That's, the key point.
So this you got from Monte Carlo.
So it's Monte Carlo, but in effect, minimizing the energy, Monte Carlo at low temperature.
So the state you have actually has, shall I say, static, stable ripples, or does it fluctuate so much that if you look at it over time?
So no, the ripple, everything we've done, our structures are always static. With everything we've done. The only thing that we are varying is the director.
So that at some point along this curve, you have an instability for the development of the non-zero AR, which you initially have no ripples.
Right.
And then you have an instability towards forming the ripples.
Correct.
And then you have the ripples for a while.
Yes.
And then eventually you get the ribbons.
Right. And the ribbons, the actual formation of the ribbons would be beyond this theory here. Because this theory just says, I throw in this rippled disk, and all I do is I study that. And so, it's this rise of the AR and the large negative Gaussian curvature at those points is suggestive of the ribbon formation. But there's not, the theory is not saying, oh yes, here, you must form a ribbon. It's suggestive, and like I say, the fact that the energy is minimized for this small value and relatively flat value of AZ is consistent with the experiment.
Bob, as for the character of the interior with that unchanging A sub z,
Yep.
You've also got the exponential decay of the
Yep.
amplitude as you go towards the center.
Correct.
Is that a length scale that characterizes that exponential something that varies as well?
So that we've just set, based on experimental observation.
And is it, was that a large number compared with the penetration?
No, that was of order, let's see, I may have put it in here. It's the B. Yeah, let's see, the B, it's small. It's again of order, I guess I forgot to put it down here. Yeah, it's of order penetration depth, something like that. Yeah.
So that not very much, in your model, not very much of the interior is actually affected.
Correct. Right. Okay, now let me show you more cool experimental stuff. As I said, the experiments are the real star here. So here's what happens if you take a disk, grab it with a pair of optical tweezers, and start pulling. You pull, and then suddenly you reach some threshold where the thing will form a ribbon, and I think the movie, then you let it go, and it snaps back. So, very, very cool stuff. So let me show you what we've done to try to begin to explore this theoretically. So, what we've done is to imagine, in some sense, a thought experiment. We're not going to look at a pair of optical tweezers. We're going to say, suppose we have, this large, and we'll approximate it, semi-infinite membrane in the XZ plane, and imagine that, we have this single tweezer that's just pulling out the edge of the membrane in some, in some localized region. And so, you could model that, a bulge by a Gaussian. We've also considered a triangular perturbation. Basically, it seems insensitive to those two choices. I should just emphasize, what we've done at this point is just an even simpler model than what I've been showing you. We don't even bother looking here at the director field. We're really just asking, will this thing want to form a twisted ribbon with negative Gaussian curvature without even thinking about chirality or any, frank energy penalty? So, we imagine something like this, and then the way, we assess, would this thing want to, as you pulled it, form a twisted ribbon, is we add a ribbon-like perturbation to the bump. And so that's what this, so now, we have a position vector of a perturbed surface. So y0 is just our original surface. That would be the y0, the yellow thing you're seeing there. And then we're adding to that this ribbon perturbation. And so in forming the ribbon, we're pulling the surface in the direction out of that xz plane, in the y direction. And then, given the tweezer experiment, we're saying that we're doing this in a localized region. So again, we've got exponential factors here. And then, what's in the red here is what will give us the twisted ribbon perturbation. And so what happened, and so then again, Professor Ouyang has a useful paper with Helfrich on the relevant mathematics. So what we want to do, remember there's no director energy in this theory. We want to look at the change just in the remaining energies, Helfrich and edge. Edge is easy. And here is what you need in order to assess the change in the Helfrich energy. You have to obviously look at the change in the mean curvature energy and also the Gaussian curvature energy. And that will come from all of this. So then what we find when we do this is the following. So these, the only difference between these two pictures is the bottom one just zooms in so you can see the structure down in this region here, which is kind of obscured at this scale of plotting. And so we're just looking at these different contributions to the Helfrich and to the edge energy as a function of the stretching, the size of that Gaussian bump. And so, not surprisingly, I mean, the edge energy is easy. The edge energy just, grows as you, the change in the edge length, just grows as a function of z naught. And then also the Gaussian curvature energy will increase in absolute value, becoming more and more negative. What may be, certainly less obvious to me, but it's easy to calculate, is the mean, curvature energy is positive, obviously. It's positive definite, but will decrease as a function of z naught. So now, what this says for the change in the total free energy is the following. So what we've done here is we've looked at it as a function of the mean curvature modulus for fixed edge energy, and also fixed mean curvature modulus and varied the edge energy. That's the difference between these two figures. And so in both cases, change in total free energy versus z naught. And the key thing is where, if at all, do these lines pass through zero and go negative? In other words, where are you likely to form a twisted ribbon? And so what you see here in the top one, so this is again for fixed edge energy, is that the mean curvature, not surprisingly, serves as a barrier to the formation of a twisted ribbon. And so if you just artificially switched off the mean, said there's no penalty at all to having mean curvature, then given the structure that we've postulated, you would immediately have a negative free energy and the thing would form a ribbon. But then as you crank up the value of k, you will then raise the critical value of the stretching needed. And similarly, if you now fix k at some value so that you don't have you have a non-zero threshold in z naught, then as you increase the value of gamma, as you're paying a larger and larger cost to form this extra edge, you will also again crank up the value of z naught, and in particular, you can reach a point where it doesn't look like you will, get any twisted ribbon at all. So I think, these are things I already said. The only thing again I should point out is this last bullet, which I think I haven't addressed yet, is, then you could say, well, how does this compare with experiment? I mean, I showed you the tweezers and they're pulling, and there's some critical value of stretching where the ribbon then forms. How does our simple theory compare with that? Again, qualitatively, yes, we say something like that, but quantitatively, we're actually off by an order of magnitude. But that again, it could be because of the simple edge model, but it could also be because we've just neglected the director field, and hence, we're not going to get quantitative agreement. Okay, so since there is time permitting, let me show you yet one more thing. This is the so-called crenellated disk. So it's first back again to the experiments. So most of what I've shown you so far has been experimental stuff on purely a particular FD virus, a particular mutant with a particular chirality. I've mentioned achiral stuff, but haven't really focused on that. But now you can ask what happens if you make achiral mixtures of left-handed and right-handed viruses. And I'll show you a movie in the next slide as to where this structure comes from. But basically, what they find when the dust is settled in these achiral mixtures is that they get structures like this, which have been termed crenellated disks. There's no phase separation. So in other words, if you fluorescently label these two different types of FD virus that are floating around here, you will find that the left and right-handed have mixed. But interestingly, what you see is, again, it almost looks like a rippling, like we were talking about a moment ago in the ripple disks. Remember, the ripple disks were just particular chirality. They were not achiral mixtures. But in this case, there are clear, even on the experimental scale, there are clear, if you look edge on, you do find the structure, even at the experimental level, rising out of the plane at these cusps. So the cusps are these intersections of these semicircular arcs. So these are the cusps. And here's a cartoon based on the optical microscopy that shows you what the FD viruses are doing. And maybe at this point, actually, let me show you the movie first, and then I can come back to this picture. So here's the way these things form. What happens is they start out with disks of opposite chirality, let's say left-handed and right-handed. And remember, this is something I showed you earlier, that when you have left-handed, it has to twist this way. When you have right-handed, it has to twist that way. But then these things will tend to coalesce because of the depletion interaction. And then you can ask, well, how does this left edge meet this right edge? And so what happens is, you form these, like I said, what we're now calling crenellated disks. But let me back up now, because now I think you'll be able to make sense of this edge-on view. If you, look at the rods here, and then look at the rods there, here's a cusp, here's the next cusp. You see that what's happened is you've tried to match a chalk. Yeah. We've tried to match a twist going like this with a twist going like that. And one way to accomplish that is just to let the surface rise up. So if you let it rise up, then you can make, these guys approach a parallel alignment. And so, that will give you a rise this way. But then if you go from, right back to left, you will actually then want to drop the surface to, again, accomplish this parallel alignment. So here again, Bob had a very nice mathematical idea of how you could model this. And so, we make a model with this structure, with this idea, and then let me just kind of, quickly show you some results. So then again, just back to the program I mentioned earlier, study the free energy as a function of these geometric parameters and see what you get. And so, what we find is that, the theory with this assumed structure predicts a non-zero AZ, it predicts a non-zero AR, and you can understand where this is coming from by kind of dissecting the energy. And basically, you've got competition here. Just as you saw in the ripple disk, you're going to be increasing the edge as you make these structures, rise in the Z direction or bulge out in the R direction. But while you're doing that, you're gaining Gaussian curvature energy, negative Gaussian curvature energy, and then there are going to be various things going on with the Frank energy that are indicated here. The net result is, here's a phase diagram where we look at this assumed structure and we compare it to a flat disk and find regions of metastability and stability. Unstable here refers to other structures that could have even more negative Gaussian curvature. What they are is, again, beyond what we've explored in this model. So at least it gives you some sense that this kind of structure could be, at least in this region here, a possible stable structure. This again, I wouldn't even pay too much attention to the numbers, and I think it's not even clear in the experiment. They've yet to somehow measure this k bar, this Gaussian curvature modulus, so it's not even clear, where one should compare this with experiment. It's really more just, a phenomenological prediction. So finally, let me end on time then. So let me kind of summarize, the main gist of what I've told you. So, what I hope you've seen are these incredible self-assemblies of FD virus that have been seen in Dogic's lab. And, the key thing being adding the polymer depleting to give you attractive interactions, also paying close attention to monodispersity. That turns out to be critical to form these structures. And then, what we think is going on the basis of all of this theoretical analysis is that these systems seem to have a preference for negative Gaussian curvature. In other words, that positive k-bar seems critical. And, at least in the chiral, systems, the molecular chirality also plays a role, leading to these twisted structures that I've shown you. And then, the idea of the theory has been to use a relatively simple phenomenological model and try to make some sense of these incredible observations. So, thank you very much.
Thank you, Bob, for a wonderfully clear exposition on a very, very beautiful system. Lovely. Do we have questions, more questions from the audience? Tom?
So you added a Gaussian curvature squared term.
Right.
Presumably it wouldn't make much difference if you just added a mean curvature to the fourth term, or would it?
Well, see, mean curvature wouldn't help me with twisted ribbon, because there the mean curvature is identically 0.
I see. Right.
So there, we had to do something.
Okay.
Right. Yeah.
And then for the crenellated things, you focused on the boundary.
Mm-hmm.
Presumably there's been some mixing of the two chirality and the bulk. Did you?
So what we did in our model, I probably didn't get to explain all the details. So again, as I said, experimentally they say these things just, mix. There's no phase separation. So what we did then in our model, I'm sorry, I should have mentioned that, is we set Q equals 0.
Mm-hmm.
So we just, shut off. They said, no. We were first playing around with all sorts of chirality fluctuations, and the experimentalists were screaming at us. So we just set Q equals 0.
So their crenellated stuff is a memory of the. Why doesn't it, if it's not chiral, then just figure out how to turn around and be flat?
You mean at the edge?
At the edge, yeah.
But I guess it would have to be a big. So you're saying once they come together.
You've tossed out the chirality. Why don't
right.
they just flip over and all go the same way?
At that point, so I guess so I know the thing is when I've seen these pictures of when they fluorescently label these things, it's I think like even near the edge it's gone. So you're saying why isn't maybe there should be some phase separation at the edge? Let's say, is that what you're imagining?
Well, I mean, if you've told me that effectively the bulk is not chiral at all,
right,
then why should the edge be, and then why not just get rid of the correlation and have all?
But I imagine there's some barrier, because remember when these things are coming together, they are chiral, and they're joining, and I think somehow in that joining is when the crenellations form.
It would only lead to crenellations where they meet, and the rest of the disks where they're not meeting, wouldn't have that imperative.
That's another question.
No, but it's still going to be rounded over at the.
No, but I guess
Right.
what Mark is saying is that you would have two crenellated points.
Oh, no, okay, so I think the confusion may be like, why did I have, I don't know, 5, right. So the point that, I understand the confusion, is for the movie I showed you, there should only be 2 cusps. Maybe that's what I didn't make clear. Maybe that's where the confusion is.
Okay.
But they do find, I don't have a movie of that, but they do find where there are multiple things coming together and they get many cusps.
Oh, I see.
Yeah, I think that was the confusion I didn't make clear. Okay.
Epifanio, you had a question.
Yes, concerning your ribbon model.
Yeah.
And I think you can actually, using the model, extract the membrane stresses associated with that energy in the deformed configuration.
I'm sorry, you want to extract the
Membrane stresses.
the flat membrane. Right?
No, the
That's
membrane,
right.
even in the curved.
Yes.
So you can just explore the stresses on the membrane in the deformed configuration. And I wonder whether when there is any line inside the ribbon along which such a stress would vanish.
So let's see, these things are fluid membranes, and so you're imagining.
There, so you can always extract distributional stresses from the surface. You imagine a cut.
Yep.
And then you ask yourself which kind of distribution of forces along the line would be in place in order to hold that degree of the deformation. And sometimes, of course, these can be derived from your energy, but sometimes locating on the membrane the regions of lines where there is the least membrane stress might help. My conjecture would be that there might be a line on the ribbon of zero stress.
And that would then mean, I'm probably not following what that would tell you then?
Kind of hinge along which you might imagine that the system is twisting.
Okay, you have to think more about that.
Dasha had a question and then
A
Buddha.
question related to this thickness of these ribbons. So you are saying that it's somehow connected to the chirality, to the pitch. You mentioned that this ratio has something, and
Right.
is something what for this? Is it a constant ratio, or is it influenced by anything else, or what?
So that ratio, like I say, it's certainly influenced by, I think, all the parameters probably play a role there. I think, we didn't explore the effect of chirality, but we certainly explored the effect of the K-bar or K24, and also the edge energy. So I think they're all playing a role. I don't have the figures here right in front of me, but they're all playing, the ratio will depend, I think, on all of those parameters.
But when you have this transition from the disc to this ribbon, so that they start to grow out, so that means how many they will start will certainly depend then on some kind of diameter of your disc, because this thickness of particular ribbon somehow is determined in a different way. So it's not just that you can have thinner or thicker, but they are some kind of natural thing.
Right.
And then depending how large is your object, then a certain amount of these ribbons will appear.
Right.
Is that true?
I think so, because I think, again, the in that movie, I would expect that the pitch to width ratio there should depend on what the dextran concentration is, what the K-bar is for the system that we don't know, and what that depends on, and that, I think, would then determine that ratio. But then you're saying how many starfish, how many arms would you
that
get?
should somehow, if
Yes.
there is
Yeah.
a preferred
Yeah.
ratio, then somehow it should depend on how big
Right.
is the circle.
Yeah, so we haven't explored any of that, because even within that simple model I showed you, we hadn't even gone. We said, we see a hint that ribbons could form, but we haven't actually even looked at the ribbons there.
Buddha had a question.
Yeah, just a few questions. So, the membrane calculation that you do, this is an achiral membrane, and there's no term like the Hellfish Pro term, which is
No, this is Hellfish Pro.
Oh,
so
okay.
buried in heat, it's we are really doing Hellfish Pro.
Okay. And the other question which I had was, when you see this, if I may call it the bleb or the ripples as you call it,
Yep.
and then things are twisting. And
Yep.
is it, is the chirality sort of conserved in this process? I mean, it's sort of because when you switch it back,
Right.
then you get an achiral mixture. So you should get
No, those are chiral. Yeah, those were not achiral.
Oh, okay.
Yeah, I realize it gets confusing between chiral and achiral, and I was confused for years myself. So, but yeah, no, those were chiral. Because I think, yeah, to the best of my understanding, if they have achiral systems, they do not see twisted ribbons. Yeah, so those are chiral.
Another question here.
Hi, the crenellations on the crenellated disc,
Yep.
do they form due to attractions between similar handedness viruses, or is it more due to inhomogeneities in the concentration of different types of virus in the disc?
I mean, it's more in just the really the depletion interact. Just the tendency for these things to want to.
Yeah, but for example, we've got on the left-hand side, we've got one type virus of a particular type.
Yes.
On the right-hand side, we've got another of another type. Is this due to an attraction between similar handedness viruses, or is it because the crenellated disc was formed by mixing two discs together? So we are creating inhomogeneities in the virus concentration overall.
Inhomogeneities in the chiral concentration.
Yes.
Yes. Yeah. So again, my understanding is that these, once these things have formed, they are, once they form, there's complete mixing of the opposite chiralities. But I think these, what's happening here is. So I guess I probably can't give you a good answer. I mean, I don't understand, frankly, the time scales here. In other words, these things form, and you could imagine how the cusps, the crenellations would form to reduce this mismatch. But then, what's going on beyond that in terms of how the ultimate mixing of the different viruses occurs, that I just don't know.
How are the crenellated discs formed? Are they formed by merging two?
It's what you saw. I can show you the. So it's this. So those were left. It's what the cartoon indicates.
Yeah.
And what you're not seeing here is any fluorescent labeling of the viruses. The only pictures I've seen are kind of when all is said and done.
So could it be that as the virus is mixing, we actually see the crenellations reduce even further because it's becoming more homogeneous? So we're getting
that over time, right? So that's a good question, and I don't. The only thing is this: I don't think so, because remember that these things, chirality or not, they want to round over.
Right.
And so I think you form this edge here. And this could have even been achiral, and there's one disc that happened to go this way, and one disc that happened to go that way. See, I don't know if they've actually tried, it may not be so easy to do, but I think you could imagine that achiral, one this way, one that way, and they get together, and they form this to reduce the mismatch.
I suppose my final question would be, have they tried, before depleting the sample, have they tried mixing the two handedness viruses together to create a racemic mixture before depletion, and then seeing what happens then? Because then you would have a nice homogeneous concentration, and you can deplete it and then see what happens.
Yeah, I don't know. Yeah, that's a good question. I don't know if they've done that. Possibly, but yeah, I'm not sure.
Are there other questions? Yes.
Do you think it's possible along with this, is it possible that you're viewing local minima so that these crenulated discs?
It's possible. Yeah. I can't rule it out. Right, exactly. That in the long run, right, something will happen. I mean, the only reason I don't think it's likely is you would have to somehow, you'd have this whole stretch of edge that you would then have to rotate the other way. And I don't know what the barrier would be for that. That's why I tend to think they're going to be fairly stable, but yeah, I'm not even sure experimentally if they know how stable
Can
they
you
are.
heat it up like crazy and then cool it down?
Right, now that I could imagine. Right, yeah, if you heated it up, I could imagine then something might then relax. Yeah, but again, there's nothing. We can't say how stable how globally stable this minimum is.
Oh, sorry. Sorry.
Well, you go ahead.
Okay. So I was interested in the formation of ribbons under quenching. Is there a phase diagram of how many ribbons under different conditions?
Not that I know of. I mean, that's the only movie. So we haven't done any theory yet that would address that. So this is kind of, I think, coming back to Dasha, what you were asking, right, in terms of just how many of these starfish arms you get. Yeah, so I don't think they've explored that much experimentally, at least not that I've seen.
Tim, you have a
Yes,
question?
whenever you get biological material producing elaborate structure, you want to ask the question as to whether or not there is biological function related to these biological structures. And you didn't say what these, what kind of cells these viruses prey on and so on, but can you comment on those things?
I can't, and I think the only thing I can say in terms of, like, where the tie-in here is with, biology and how it was, sold to the NSF or the MRSEC is.
This is being recorded.
I know, I'm thinking of as I'm saying that, why am I doing this?
CIA have already put
That's right. The NSA is now.
Steve can edit these.
That's good. Is really more the idea is to explore concepts of confinement and frustration. And that's really, where this kind of was viewed as a model system, but not particularly with, what kind of bacteria does FD attack. Yeah, that was, not, I think, part of the deal.
I can answer that. You're asking what bacteria attacks?
Well, I was asking the question that came after the question of what kind of FD they attack, which is these are really quite elaborate structures, and the elaborate structures might have something to do eventually with evolutionary fitness of these particular viruses under different kinds of circumstances. And I just wondered whether.
Yeah, and I think the idea was more, like I say, kind of conceptual, in the sense of, if you're interested in cell structure, you're interested in confinement, you're interested in chirality, you're interested in frustration. So here is, this system where you can explore these ideas in some, general conceptual
Do
way.
tell us which bacteria it does prey on.
It's E. coli bacteria. It has to possess an F prime factor, which is essentially a pilus it would use for sexual reproduction, and it basically attaches onto the pilus, and the bacteria attracts it in, and that basically allows it entry into the cell. It's non-lytic, so it doesn't kill the bacteria. It just sort of gives it a prolonged cold, which means that under stress conditions it just produces lots and lots of it. The original strain was called M13, and I think it was found in a sewer in Munich, hence M. But they've also found that by changing a couple of base pairs in the genome of the main coat protein, it actually becomes chiral, because the initial M13 is just pneumatic in phase. But just changing a couple of the amino acids on the main coat protein has somehow broken the symmetry, and we've now got these chiral structures, which is
Yeah, and even the left-handed versus right-handed chirality, from what I understand, is one amino acid in the coat protein.
It's really amazing.
It
how
is
it can
amazing.
change.
Yeah.
So we like these guys because they're against E. coli, and E. coli are sometimes dangerous.
Well, sometimes dangerous. It has been, basically, they're indiscriminatory, really, when it comes to what sort of species of E. coli. Basically, as long as the E. coli has one of these sort of like sex organs that sometimes they have, it will attack it. But it has, I think, I have heard that it has actually been found to cure diarrhea in piglets. So it has actually improved the mortality rate in farms in some cases. So it does have some benefits to humans.
I couldn't help noticing in your film that the two droplets that were coming together were of a different size, and therefore maybe the final mixture is not racemic.
So in that one, right, possibly. Yeah, I'm trying to think. The pictures I've seen where they've actually done the fluorescent labeling, right, no, that's certainly very possible there. Yeah, and I'm not sure that if the fluorescent pictures, happen to be exactly what you saw there.
Are there any other questions? If not, let's thank Bob again.